Swayam Prabha. Digital India. Educated India.
Hello, my dear students. Course title Medicinal Chemistry. Lecture title Physicochemical Principle of Drug Action. Part 2. Faculty name Dhamak Vikrant Murlidhar. And module number 4. Lecture number 7. So in the last lecture also we have seen physicochemical principle of drug action. And so remaining part now we'll conduct in this lecture. So there are some physicochemical principles of drug action. Earlier we have seen like partition coefficient, dissolution, pKa, solubility, and now in this lecture we will be looking stereochemistry. After stereochemistry we will be looking electronic structure and Hammett correlation, relationship between chemical and biological data, and we will be looking by Hunt's approach. So let's begin with the first content of today's lecture, that is stereochemistry. The stereochemistry involves the study of three-dimensional nature of molecule. So whatever the structures we are looking in the chemistry, so these are two-dimensional structures, or whatever it is written on the board or paper. So these are nothing but two-dimensional structures. But in actual, these are three-dimensional structures. The isomer which have same bond connectivity but different arrangement of groups or atoms in the space are termed as stereoisomers. So here isomer, the meaning of isomer, it is that it is having same formula, structural formula, but different physicochemical properties. So the meaning of isomer is that it is having same number of atoms or same number of atoms in a molecule, but its properties are different. So the property in sense physical and chemical. As soon as chemical property changes, so there will be change in physical property also. So it is intercorrelated that physical and chemical property changes as the structure changes. But in case it is having little bit different, stereoisomers are little bit different. So general isomers are having two classes, and in one from that one, that is stereoisomer. So stereoisomers have same bond connectivity but different arrangement of group. So their molecular formula is same, structural formula is same, but only the arrangement of atoms in the space or connectivity of atom in the space, it is different. And this term, it is said to be an stereoisomer. So in case of an example, so this one, it is an example of an geometrical isomer, and this geometrical isomer meaning of geometrical isomer is that it is having a double bond in between, carbon-carbon double bond, and atoms attached to an carbon-carbon double bond. So now here you can see completely that similar is the structure, that is carbon-carbon double bond. Only the arrangement of an atom it is different. So here you can see it is an cis type of an arrangement. So cis, it means that groups or same priority groups, they are on the same side, and groups which same priority groups which are on different side, it's said to be a trans structure. So the from definition also it can be understood that there is connectivity, it is same. Okay? So the carbon it is attached to and similar chlorine molecule. Here also a chlorine molecule has been attached with an carbon, but only the arrangement of atom or groups in the space is different. So the meaning of this thing is that in the space it is different. That now here you can see it is not a mirror image of each other. So they differ because the arrangement it is little bit different over here. Because here carbon it is having chlorine from this side, but carbon now it is having from an back side. And these are particular in geometric isomer because these are having a double bond in between. And trans structure, it is more stable than cis structure. So this is due to an steric hindrances, because if we consider this group, so these are sterically repulsive or sterical, there will be a steric repulsion because they are close to each other. But here now the distance it is quite long, and due to which there will be no steric repulsion, or there will be very minimum steric repulsion. But in case of this one, there will be a steric repulsion. So that is why trans isomers are more stable than cis isomer. But particularly for any activity, so it does not depend that trans will only show an activity or cis will not show an activity or less activity. So it depends on interaction with an receptors, because what kind of structure does fit, or what type of key does fit in the lock or receptor, it depends on that. So as in general trans are more stable, but it does not happen that trans will show more activity. So activity depends on the receptor interaction. So now we'll see stereochemistry of drugs in actual. Drug must approach the receptor and fit closely to its surface. Take an example. If we take a drug, and when we take a drug into our body, so there are a lot many, like we can say there are a lot many locks, and drug is like a key. So it will fit only in that socket where drug it is having a particular socket, or it will fit to an such a socket where its structure resembles or its structure fits. So drug must approach the receptor and fit closely to its surface. As you know that particular drug will show a particular response, because the affinity for binding or it requires a particular socket, and so that is why it approaches a particular receptor. Steric factors determined by stereochemistry of the receptor site, surface, and that of a drug molecules are therefore of a primary importance in determining the nature and efficacy of drug receptor interaction. So drug receptor interaction, it is one of an important thing, but for that purpose one should know an steric factor. So steric factor, it means that when there is steric repulsion due to some of the groups present in a structure, so it will show either molecule it fits into receptor, but due to a steric repulsion, so the efficacy of molecule it will decrease or increase. So steric factors are important for it. So we'll see one of an example. See, this is an optical isomer. Why optical isomer? Stereoisomers are being classified into two classes. So one of an class, that is geometrical isomer, and second class, that is an optical isomer. So optical isomer, meaning of this optical isomer is that it can rotate the plane of plane polarized light, and it is, for an optical isomer, a molecule should have an chiral carbon atom. So here you can see a molecule, it is having an chiral carbon atom. So the meaning of chiral carbon atom is that it is having a different groups in all four directions. So these are all different four groups in all the directions, and this dotted line, it is indicating that this group, it is behind the carbon, because it's a three-dimensional structure which has been written, and such type of an presentations are said to be an Fisher's presentation. So sorry, not Fisher's presentation, so it is said to be flying wedge representation. So this dotted line, it is indicating that it is away from the observer, and this broad line, it is indicating that it is with the observer, or it is in front of an observer or close to an observer. So here now you can see why stereochemistry is important for drug interaction. So this is a structure of an receptor, but receptor, it is not having a particular match. So here you can see that it should have a red molecule over here, a red atom over here, and green atom on the space on this space of this red one. So then only it will match. For blue, it is correct that it will fit. So it is like lock and key, that key should fit into a lock. So now here you can see, now it matches. See, now there is difference, that red, it has come in the front, and this green atom, it has gone behind the carbon, and now it fits the receptor. So this is how the stereochemistry is important for medicinal chemistry or drug actions, because a structural three-dimensional structure of a molecule, it will determine an efficacy of drug receptor interaction. So to have a proper efficacy, or to have a proper potency of a drug, so it should fit into a receptor like in lock and key model, and that is why stereochemistry of drug, it is important. So now we'll be looking towards importance of stereochemistry. So stereochemistry plays a major role in pharmacological properties, because any changes in stereospecificity of drug will affect its pharmacological activity. So in the last slide also we have seen that stereospecificity, it is important, because molecule to adjoin and particular site, it should have a proper three-dimensional structure, so that it will fit into a receptor, and after fitting into a receptor, there will be interaction often some groups between receptor and a molecule. So receptors are basically nothing but an protein, and these proteins are made of an amino acids, and these amino acids, they are having different atoms for hydrogen bonding or ionic bonding or covalent bonding, and after bonding with an specific groups in a drug molecule, so it will either mimic the action or it will retard the action. And also stereochemistry is important because isomeric pairs have different physical properties like log P and pK, and thus differs in pharmacological activity. So in the last lecture we have seen the concept of log P and pK. So log P, it is nothing but a partition coefficient, and this partition coefficient, so half of the drug molecule, it is not half of the drug molecule, but drug molecule is solubilized into two phases, that one is organic phase and second one is an water phase. And so our body, it is composed of some membranes, and these membranes, they are lipophilic in nature, but to come towards a molecule transport towards a lipophilic membrane, so molecule also should have an ionic interaction with body fluids, because body fluids, they are ionic in nature and they will carry only a drug which is ionic in nature. So molecule should be not only an lipophilic or hydrophilic, so it should have a balance. And after that, we have seen a pK that 50% of drug it is in the ionized form and unionized form, and it is at particular pH. So pK is also important for your drug action. And as isomers, they have different physical and chemical properties. So different physical and chemical properties in sense they will be having different solubility, they will be having different dissolution rate, they will be having different refractive index, and many more physical properties will change. And as the physical properties also change, there will be change in chemical properties also, and thus it shows a difference in pharmacological activity. So in case of an pharmacological also, or in case of a medicinal chemistry also, you will be coming across there are a lot many molecules. They are different pairs of an enantiomers, and different pairs of an enantiomers shows different activity. Like we will take an example of one of a drug which is widely used in the market, that is cetirizine. So it is one of an second generation and antihistaminic drug, and which is used for some symptoms like sneezing, coughing. And for this particular symptoms, cetirizine it is being utilized when histamine it has been released, so it is an antihistaminic drug. So earlier there was only an cetirizine in the market, but later on you can see now there is only one pair of an enantiomer, that is leocetirizine or L-cetirizine. So nowadays you can see that there is leocetirizine. So developments or research was made that whenever drug it was been synthesized, so it was coming in the racemic mixture. So racemic mixture it is nothing but an equal form of an enantiomers. So 50% of enantiomer it is R and 50% that is S. So which is having an optical difference which can rotate the plane polarized light towards right and towards and left. So depending on that physical properties, earlier cetirizine it was given in a racemic form, but nowadays it was but later on it was been found that cetirizine it is having a particular leocetirizine it is having a particular pharmacological effect, that is it is having more pharmacological effect than R-cetirizine, and that is why we are now watching an leocetirizine which is being utilized. So from this you can understand the importance of stereochemistry, that for particular binding the stereochemistry aspect is very important, because the stereochemistry of molecule it will be responsible for potency and efficiency or efficacy of a molecule, and that will show an pharmacological activity. Now we'll look particular class example. So stereoisomers are of specifically two types. One is geometric isomer. So geometric isomers and optical isomers, they differ into some properties. Like geometric isomer, it is for an carbon-carbon double bond only. When there is carbon single bond, so it becomes an optical isomer. But all compounds are not optical isomer. It also has some criteria that should be fulfilled by optical isomer as well as an geometrical isomer. All the carbon-carbon double bond are not an geometrical isomer. It should also fulfill some criteria. Geometric isomerism is represented by cis and trans. So cis and trans, these are two types of an geometric isomers, resulting in restricted rotation due to carbon-carbon double bond or a rigid ring system. So rigid ring system over here it means that in case of some steroids, so it is having four rings in case of steroids, and these steroids they are having a particular rigid system because they are not able to break. But carbon-carbon double bond, when it is being heated, so at particular resistance it can be it will break and again it will rejoin. So cis can be converted to trans and trans can be converted to cis, because the carbon-carbon double bond it can break, and this carbon-carbon double bond it breaks due to a heat. and all the time a molecule it will try to attain and more stable structure, and the more stable structure over here it is nothing but a trans. Earlier also we have seen that a trans it is having a uh, different arrangement, that atoms are in the opposite directions, and in case of an cis atoms are in the same direction or on the same side, and that is why trans is more stable. But it does not happen that trans it is more stable then it will show an more action. So it all depends on how molecule it interacts with an receptor at the receptor site action, and what type of interactions, what type of groups are needed for an interaction. So all it depends on arrangement of an molecules or arrangement of an receptor over there. Like we will take an example. So now here you can see a geometric isomer. So this geometric isomer it is having a carbon carbon double bond over here, and it is now having a restricted rotation because it cannot rotate easily. So it happens in the case of an optical isomer that molecules can rotate, but it does not happen in this case because this pi bond between carbon carbon, or we can say sp2 hybridized carbon, is unable to make a rotation. When it is an sp3, so molecule can rotate, but when it is an sp2, or we can say when it is carbon carbon double bond, so one of bond it is an sigma and second one is an pi, so molecule will now is unable to rotate. It can rotate, but it requires certain energy, and certain energy will rotate it. But as in the normal molecule will not rotate easily. So it is one of a drug that is trans diethyl stilbestrol and cis diethyl stilbestrol. So here you can see that these are priority groups, priority number 1 group and priority 1 number group on carbon carbon side, and this is a second priority group. So these are opposite to each other, and also first priority groups they are opposite to each other. But in case of an cis, so they are on similar side beyond the carbon atom. So here also you can see this ethyl chain, so it is on the similar side, and here phenolic chain, so it is also on the same side, and that is why now there will be an interaction between these two, and this interaction between even in aromatic ring also, that will make molecule unstable. But here in case of this one, so these are very apart, or these are having a long distance, and that is why trans structure is more stable than the cis structure. And estrogenic activity, so when this molecule show an estrogenic activity, so it is one of an a drug responsible for an sexual hormone production. And when it comes to an cis, so cis it is having a minimum activity, so it is having only an 7% activity compared to an trans isomer. So from this you will be understanding that how important stereochemistry of a molecule is, because stereochemistry it is particularly and part of an organic chemistry, but it is very important for a drug action, because all when molecule it dissolves, when molecule it firstly when it disintegrates, after that it dissolves, when it crosses an membranes, when it comes to an receptor interaction for at the site, so it is very important what stereochemistry it brings with it, and depending on that stereochemistry only a drug molecule it will get attached to an particular receptor, or it will show an particular interaction. So now next concept from stereochemistry, that is enantiomers. Chiral molecules having non-superimposable mirror images are said to be an enantiomer. Enantiomers have different biological activity because physical and chemical property changes. So we'll take an example of an adrenaline. So this is a structure of an adrenaline, and these are the binding sites into an receptors. So this portion, it is for an aromatic ring, and this portion with the green color, it is for OH, and this one, it is for an quaternary ammonium. So enantiomers are non-superimposable mirror images. So we'll look from the structures. So here you can see both are like a mirror image of each other. This structure is now and horizontal, but if we draw a vertical structure, so you can see that it is an exact mirror image of this one, but only the difference is that these are non-superimposable. So the meaning of non-superimposable is that when S enantiomer it is kept on the R enantiomer, so that will be not an imposed on each other. So because this there is difference of in this arrangement in the space of OH and H, and also here you can see that OH it is coming towards an observer, and here it is going away from an observer, and this makes it as a non-superimposable mirror image. Actually, optical isomers they are of three types. One is diastereomers. So in case of an diastereomers, molecules are not mirror images, but in case of an enantiomers in between, so these are mirror images, but they are non-superimposable because they are not imposed on each other. And third one, that is mesocompounds, and mesocompounds are such compounds that are mirror images with an superimposable chemistry, because these can be imposed on each other. And mesocompounds are optically inactive because the rotation, whatever it has been caused because of this chiral center, it has been nullified by each other, and that is why the mesocompounds are optically inactive and enantiomers are optically active compound. And also with that, a molecule should be an chiral. So to be, and so any molecule which is a straight chain structure or which is having a different groups on the carbon, it does not mean that it is an enantiomer. For an enantiomer, firstly it should possess an property of that molecule should be an chiral one. Once it is chiral one, then only we can say it is an enantiomer. Because when molecule it is chiral, so the meaning of chiral is that it is having an 4 different groups. So here you can see it is there are 4 different groups: H, OH, aromatic ring, and this quaternary ammonium with an methyl in between, a bridge, and due to which it makes an chiral. If suppose there is a hydrogen in place of this methyl bridge with an quaternary ammonium, so it will not become an enantiomer because it loses an property of an chiral carbon, and this chiral carbon it is responsible for an optical rotation in case of an stereoisomers. But this rotation, so we can say now these are different conformers. So conformer, it is nothing but an rotation around the carbon. So here you can see that now there is difference between an angle, or there is difference in arrangement, and due to this arrangement we can say, so this is a different type of an conformation, and this is a different type of a conformation. So the conformation, it means that there is rotation around the single bond, the carbon joined single bond, and due to rotation it forms an enantiomer. So this R enantiomer, it has an high binding capacity compared to this S enantiomer, because here you can see that OH it is not available for an binding now, and here it is indicating that plus sign over an nitrogen. So it is indicating that nitrogen it is having, and so it is an electron deficient center because it is having a plus sign, and the receptor with which it has an interaction, or the amino acid with which it has an interaction in the receptor, so it is an electron rich center, and it will donate an electron towards a nitrogen and it will form a bond. But in case of this one, OH, so OH it does not have an OH, it is an electron rich center, and it does not need an electron, because already it is an electron rich center, but on the receptor here it is an electron deficient center. So these are important aspects where it is an electron rich center and where it is an electron deficient center. Sometimes they are said to be an cationic site and anionic site, cationic positive charge and anionic negatively charged. So these are called cationic sockets and anionic sockets. And here comes an aromatic ring. So aromatic ring, it does not have an interaction with a receptor, but it is need like an pocket or to fit into a receptor, or we can say here it might be an lipophilic pocket, and as aromatic ring is also lipophilic in between, and this lipophilic pocket will fit over here. But there are some groups adjoining with this aromatic, that is OH, and these are responsible again for an interaction with a particular receptor at the site. And due to structural difference, or we can say structure difference in stereochemistry of a structure, that position of an OH and connectivity of that OH with the carbon, it is different in the space, and that is why there is difference in the binding. And here binding, it is different. Only two groups or two atoms, they can only bind to a receptor. But this OH, it is not available for binding, because now it has gone behind the carbon atom, or it is in the plane of an carbon atom, but hydrogen, it has come in front of an observer. Now we'll be looking towards an next concept, that is electronic structure and Hammett correlation. So till yet we have completed the stereochemistry part. Now we'll be coming towards an electronic structure. Electronic parameters mainly indicate the influence of polar characters of drug on its biological activity. They affect metabolism, elimination pattern of drug, and drug receptor interaction. So in earlier case also, we have seen that in the last lecture, that ADME, that is pharmacokinetics, absorption, distribution, metabolism, and elimination. Electronic parameters are important for its biological activity, so because as polar characteristics increases, so lipophilicity of a molecule decreases, and as the lipophilicity of molecule decreases, so there will be so the molecule will be less available for metabolism, as absorption from a lipid bilayer or lipid membrane, so it decreases, and as it decreases, so there will be less distribution into a tissue compartment, because they allow only a lipophilic molecule to enter. But as the polar characteristics, or we can say when molecule becomes more polar, so it will lose activity, or it will get dissolved into an aqueous medium into our body, and it will be directly been eliminated from our body, rather than it has been metabolized, or it is available for an interaction with a receptor, so it will not cross and barriers over there. And polar activity, as earlier also we have seen that when molecule it becomes more polar, so it will get eliminated. When molecule it is more non-polar or more lipophilic, so molecule it will stay in the body for a longer time. But as molecule will get polar, and this polarity it comes from a metabolism, because in case of a metabolism there is alteration in structure, or metabolism it causes introduction of some polar atom into a structure due to some enzymatic activities like cytochrome phosphate 450, and lot many enzymes, like there will be certain reactions like oxidation reductions or catalysis, and this will cause this will cause change in the structure, and this change in the structure, it is desired one that after metabolism, so molecule should become an polar, and as molecule will be becoming an polar, so it will show and polar characteristics, means it will show an ionic group on it, and as ionic group it interact, or it will get dissolved easily into an body fluids like water, and it will be get eliminated. To eliminate a molecule, it should dissolve in the water, and it can be eliminated by certain ways. It may be sweat, it may be facial, it may be urination. So there are certain ways to eliminate. And this Hammett correlation, so this Hammett correlation explains how what type of an group these are responsible for molecule making more polar. And so Hammett postulated that electronic effect of substance in different organic reactions should be similar. So Hammett has postulated that so electronic effect on the molecule, it is due to an substance. So substance, so he has taken an example like an benzoic acid, and this benzoic acid when it is not been substituted, so there will be difference in ionization. So particularly this Hammett correlation, it explain about an ionization. So what effect does an substituent makes on an ionization of a molecule? So he considered a standard benzoic acid as a standard, and after that what are the substituents, or how a molecule becomes polar, or how molecules loses its polarity or it becomes a non-polar. So it depends all on the substituent. So whether these are substituents are electron withdrawing or electron donating, so that it is been correlated by Hammett. The most commonly used parameter in Hammett substituent constant, that is sigma, which is obtained from dissociation constant. So we have seen this particular concept of dissociation in case of an pK. So it is nothing but when acid it dissociate from an acid form, so it forms an conjugate base, and so the concentration of this K value, or we can say concentration of acids and its conjugate base, so it is nothing but an dissociation constant. And dissociation constant, so dissociation constant, it increases as the ionization increases, because molecule will increase ionizing property because of an polarity. As the polarity in the molecule increases, so there will be an fast dissociation, and due to which the value increases. So here in case of an Hammett equation, sigma has been utilized as an substituent, and so from this it can be understood that as what is the effect of an dissociation constant on and biological activity. So either biological activity it increases or decreases. So it is not in the particular direction whether the value of sigma it increases and biological activity also increases. So it is depend on what type of a desired action we require, and where molecule it interact with the receptors, and so that is important. And this constant sigma constant, which is important, and it has been explained by an Hammett. And this Hammett correlation is also important in case of a drug designing. When a molecule of series it has been designed, so what are the effects of an electron withdrawing groups, and what are the effects of an electron donating group on particular substrate or particular molecule? So that will decide the biological activity of molecule. If suppose in the particular direction, if electron withdrawing group increases and biological activity, so the researcher makes in a such way that it should have an electron donating group only on it, because as the biological activity will increase. So this was just an example how researcher makes a series of an molecule. So Hammett correlation, it explains an electronic properties of a molecule, And there are actually lot many things to explain in Hammett correlation, but for our purpose, we just are stick only towards an electronic parameter, that we can understand that Hammett correlation, it explains an electronic parameters which are present on molecule, and what are the effects of this electronic, or how this electronic structure it influence and biological activity. So we are limited only up to this concept. Actually, the Hammett correlation in case of an organic chemistry, it is quite wider. And when any synthesis it has been when any synthesis of a molecule it has been designed, so in lot many cases Hammett correlation it has been utilized by a researcher to understand ionic properties of that molecule, or easily ionization of a molecule. So particularly from this thing, one can understand that polarity of a molecule, and how polarity of a molecule is important for a biological activity, or it influences a biological activity. But Hammett correlation is such a thing that it will make you understand. So what are the how electronic electronic parameter it increases, or electronic surface, or electronic we can say electronic environment of a molecule affects and biological activity. So now we'll see a relationship between chemical and biological data, that is Hunt's approach. And it is also said Hunt's analysis. So Hunt's analysis is the investigation of quantitative relationship between the biological activity of series of compounds and their physicochemical substance or global parameter representing hydrophobic, electronic, steric and other effects using multiple regression correlation methodology. So these are important things for a drug designing, because see, like an example, so here it has been told that a series of an compounds it has been prepared. Whenever there is, we can say a drug, whenever it comes to a market, so it does not directly comes to an market. It has to face lot many things like clinical trials or preclinical trials. But before that drug, it has been synthesized in a series, and this series it so this series uh, it will help to produce more and more molecules, and a little modification between a molecule, a series of a compound it has been prepared, and from that series an active molecule it has been isolated. So this isolation of an active molecule, it depends on a biological activity of that particular compound from that particular series, and all it depends on physicochemical substance. So physicochemical substance uh, physicochemical substance means groups which are substance which are responsible for change in physicochemical properties. So Hunt's approach, so it has particularly looked upon three main parameters. So one is hydrophobic. When there is introduction of an lipophilic groups into and molecule, so it will increases an lipophilicity or hydrophobicity. And when there is the electronic parameter, also we have seen an Hammett correlation. So when there are polar characteristics of a molecule, so it has been explained by an electronic. And lastly, it is nothing but an steric. So earlier also we have seen in case of an stereochemistry that how steric parameter it influences. So these three parameters Hunt's approach has discussed. With a series of a compound, this what changes does it takes place. So these are important parameters for quantitative structure activity relationship. Shortly, it is said to be an QSR. So now we will be it has been best. So Hans proposed drug action depending on two processes. Firstly, the movement of drug from the point of entry into the body to the site of action, and secondly, the interaction with the receptor site. Hunt suggested linear and non-linear dependence of biological activity on different parameters. So these were the two proposed things by Hunt. That is, firstly, the movement of drug from the point of entry, means where from drug it has been administered, and to the site of action. So movement, it does require a lot much. The drug should pass through and lot many membranes, and depending on that, Hans proposed an postulate. Secondly, the interaction with the receptor site. Once drug it is moved from the point of entry, means from point of administration, towards the site of action. So secondly, it should interact with an receptor. And on the basis of these two, so he made two equations. One is linear and second one is a non-linear one. So the linear here, it means that biological action, it increases linearly, and as the concentration increases, there will be an linear increase in the biological action. But one of an thing, he made an equation, so it was based on non-linear dependence. So the meaning of this thing is that some parameters, they are independent, and due to which it shows an parabolic pathway, and we does not get an linear relationship. Now we'll be coming towards one of an dependence, that is linear dependence of an biological activity. The correlation of biological activity with physicochemical properties is often termed as extra thermodynamic relationship. Biological activity within the homologous series of compounds to set of theoretical molecular parameters which are assumed to describe essential properties of drug molecule. So earlier also we have discussed that based on the three parameters, basically. So these three parameters, these are hydrophobicity, means lipophilicity we can say, and next it is electronic structure, or it is correlated with an Hammett correlation, and lastly it was steric effect. So these are the three parameters. And when homologous series, so earlier also I have discussed that when a compound it has been developed, so it has been developed with an homologous series. So making some changes with an molecule of an small molecules, removing or inserting some chains or some molecules which are having an electron donating and electron accepting capacities, or it may be an lipophilic region. So depending on that, the homologous series it has been designed. And so essential these three parameters: hydrophobicity, electronic structure, and lastly steric parameters. So biological action depends on all these three, and so by increasing lipophilicity, there may be increase in linear relationship with an biological activity, or increasing in electronic or increasing in polar capacity of a molecule. So there may be an linear dependence, or if we some molecules or some atoms, these are responsible for some steric entrances. So there may be an linear dependence. So it may be either increasing or decreasing one, but it is linear one. So it is an equation for an linear dependence of biological activity. So where C is a concentration of drug producing the biological response being measured, and the log of 1 by C it has been taken. After that, log P, that is substituent constant for solubility, so it is referred as pi. So earlier also we have discussed what is log P in the last lecture. After that, ES, that is Taft constant. It is for steric effects. And after that, Hammett substitution constant. So these three are important parameters, that is lipophilicity. So it refers to and how does the lipophilicity changes with an substance, or solubility changes with an substance. And after that, steric factors or steric effects, and after that, Hammett substitution constant. So electronic structure of a molecule. And this ABCD are the constant of a system which are determined by a computer to obtain best fitting line. So from this, one can understand, from this equation, we get a particular value, and that particular value, from that particular value, one can understand. So at what optimum value should biological response be, or biological activity it is at what particular value. And so the drugs are designed in between that only, so that it will show an uh, specific action or desired action. Now after linear one, so there is one more concept, that is non-linear dependence of an biological activity. Increase in log p value from log p0 does not linearly causes increase in biological activity. Sometimes it decreases. So the meaning of this thing, as lipophilicity increases, so it does not mean that biological activity, it increases linearly. So sometimes it decreases and it shows an parabolic equation. Hunt suggested that the compounds could be involved in random walk process. Low hydrophobic molecules had a tendency to remain in first aqueous compartment, whereas highly hydrophobic analogs sequestered in first lipoidal phase that they encountered. So when there is entry of a molecule, so it may be in a so the random walk, it means that molecule with low hydrophobic character, so they will remain in aqueous compartment. So low hydrophobic, it means that so it is inverse to an hydrophilicity. Phobic and philicity. Phobic means it will cause a repulsion, or lipophilic character it will increases, and hydrophilicity, it means that there will be a low lipophilicity, so it means then aqueous character increases. So the molecule, it will remain firstly in aqueous compartment, whereas highly hydrophobic analogs, they will be firstly come into an lipoidal phase. So hydrophobic analog, it means over here, so it is nothing but an lipophilic molecules, so they will come into an lipoidal phase easily, because lipids, membranes are an lipid bilayers, so which allows and this hydrophobic molecules or analogs to come easily into it. This led to a formulation of a parabolic equation relating to an biological activity and hydrophobicity. Now we'll see an equation for this non-linear one. And also in the random walk process, the compound partition in and out of various compartments and interact with the myriad biological components in the process. So lot much of compound, it is also bound with an proteins, and due to which compound it has been lost, and the actual dose of a drug, it does not reaches any time at towards an site of an action for an biological response. Non-linear dependence of biological activity. So Hans has developed an equation for a non-linear dependence. So non-linear itself indicates that so there is no relation with an hydrophilicity or hydrophobicity, or we can say the steric factor or electronic factor. So it does not depend directly on biological activity. So there is an parabolic pathway, or there is a curved graph obtained for an biological activity. So P that is partition coefficient. So partition coefficient between octanol water. So octanol, it is used for an in vitro measurement. Sigma, it is Hammett electronic parameter. A and B are regression coefficients. So A and B are regression coefficients. ES, similar as it was in linear one, that is steric factor. And K, constant term. So these are the two new terms for a non-linear one, that is rho and delta. So rho and delta are proportionality constant, designating sensitivity of reaction to an electron density. So again, it depends on the electronic parameter. And this equation, it is little bit different from a linear one, because partition coefficient here it has been squared. Significance of Hunt's analysis. The Hunt's correlation pieced together is valuable information of newly designed drug molecule in more plausible and predictive, quantifiable manner than before, and apply it to biological system more logistically and judiciously. So once we get an information of an lipophilicity, that is hydrophobicity, or electronic parameter, and lastly an steric factor. So from that information, whether at which particular instance or by increasing such parameter does activity increases, or by decreasing that parameter activity increases or decreases. So logistically it can be identified, and this studies Hunt's analysis, it is used particularly in studies of an QSR, that is quantitative structure activity relationship. This particular concept further sustained and expanded by assuming all these substituents. So already we have discussed these things, that is hydrophobicity, electronic parameter, and that steric factor. So so it will govern the potency of a drug molecule and also an efficacy for binding with an receptor. So these three are important parameter to show an potency as well as an efficacy of a drug substance. So Hunt's analysis, it is important in QSR analysis. Now we'll see the summary of this lecture. So particularly we have seen three physicochemical principles of drug action. So out of them, one is stereochemical nature of a drug. So we have seen an example within 3 dimension of drug receptor interaction. So particularly drugs should have and a structure which should fit into a receptor, and that 3-dimensional structure we have seen how does it fit, and which are the how if compound it does not show an action, so what is responsible for its minimum action or low action. So we have seen how the drug receptor interaction it is. After that we have seen one of an stereoisomer, that is enantiomer, with an example, that is R adrenaline is more active than S adrenaline. So enantiomer it does have a different conformer, and this different conformer changes the connectivity of an atom in the space, due to which there is difference in the action. After that we have seen one more stereoisomer. So these are the 2 types of an stereoisomer. One is geometrical isomer and second one is an optical isomer. So optical isomers, in that we have seen an example of an enantiomers. So geometrical isomers, like we have seen an example, which are double bonded carbon. Cis diethyl stilbestrol shows only 7% activity compared to an trans form. So it is due to an binding with an receptor. So this binding with an receptor, it is having a different conformation and different arrangement, due to which cis is less active than the transform. After that, we have seen electronic structure and its effect. So electronic structure or the polar groups in the molecule causes influence in metabolism, elimination and drug receptor interaction, and also which affects an biological activity of a compound. After that, we have seen structure activity relationship by physical and chemical means. So that is nothing but an Hunt's approach. So we have seen a linear and non-linear dependence of biological activity with and equation. Thank you. So we have completed module number 4 and lecture number 7.